Good evening, viewers. Today we are going to talk about Human Genome Project. So welcome back to my channel again, and if you haven't subscribed as of now, please do it as early as possible because you won't miss any of these videos. Human Genome Project. It formally began in October 1990 and completed in April 2003. It continued for about 13 years. The project goals. What were the goals of this project? Number one is to determine the base pairs that make up a human DNA. Number two, to store all the informations in the database. Number three, to identify 25,000 genes approximately in human DNA. Number 4 is sought to address the ethical, legal and social issues ELSI. Number 5 to molecularize diseases and their possible cure. Number 6 to improve tools for data analysis. And number 7 to determine the sequences of about 3 billion chemical base pairs. Actually, this project sequences only euchromatic regions of the genome, not the heterochromatic region. What are the methodologies? There are actually two methodologies. One is known as EST or expressed sequence tags, which focuses only RNA expressed genes. The another one is sequence annotation technique. It focuses on both coding and non-coding sequences of the gene. Now, in March 2009, the Genome Reference Consortium released a more accurate version of the project. The methods involved: number one, isolating total DNA from the cell and breaking into random fragments. Number two, the fragments were amplified by cloning using bacterial artificial chromosome and yeast artificial chromosomes. Number 3. Sequencing amplified fragments using automated sequencers. Number 4. Arranging sequences on the basis of overlapping regions. Number 5. Alignment of these sequences. Number 6. Annotation of sequences and assigned to each chromosome. And number 7, assigning the genetic and physical maps on the genome. Actually, these things come during the time of setting questions. What were the steps involved during DNA sequencing during HGP? Now coming to Rice Genome Project, RGP. This was done by International Rice Genome Sequencing Project in September 1997, that is IRGSP. The reasons for this project: it is staple food for more than half of the world's population. Rice is widely used. Number 2, rice has the smallest genome size among grasses, like just approximately 430 MB. Rice shows high diversity. Rice easily transforms genetically, and it has collinearity with other cereal crops like wheat, maize, sorghum, etc. Now, applications of the project. Number 1, it helps to enhance molecular products. Number 2, the large varieties of mutants of rice manufactured artificially. Number 3 is IRGSP provides excellent resources for other eukaryotic genome sequencing. Number 4 is it provides valuable info on evolution of plants. Now, what are its major findings? What did we get actually? Number 1 is protein identification resources database found significantly among 25% of rice sequencers. Number 2 is accuracy and reproducibility of linkage analysis based on RFLP. restriction fragment length polymorphism is higher than other methods. Number 3 is mapping revealed that chromosome orientation of each linkage group was with short arm on top and the long arm at the bottom. Number 4 is at least 53 of the bacterial artificial chromosomes have been annotated by it. Now what about findings of HGP? Number 1 is the biggest known human genome, human gene dystrophin, which consists of about 2.4 million bases. Number 2, we have found out the chromosome number 1 contains the largest number of genes, that is 3168, and the Y chromosome contains about 344, the smallest number of genes. So that's all for today. Please like and share as widely as possible. Thank you.